This is a Dendrochronology lab. It's established in 2013. And Dendrochronology means, the word comes from dendros, means trees, chrono means time, sequence, events, and ology is study of. So Dendrochronology is the dating and study of annual rings in trees. Dating trees doesn't mean just counting rings. It's more than that. Even in temperate regions where they have this distinct climate seasonality, like winter and summer. It has been used for so many purposes in the last century. Unfortunately, in the tropics, there was a misconception that trees do not form rings. But during the last few decades, we have proved that there are many trees that form rings in the tropics. Basically, we collect samples from living trees using increment cores. And also, you just have to go to the field and you use the Swedish borer. You press to the wood and you core sample and you bring it to the laboratory. The other is collect stem discs using saws, and it can be from dead trees or stumps or logs. And we collect that to the laboratory and we measure different parts of the wood material depending on what we want to know. It can be the wood anatomical structure, it can be the ring widths, or the chemical composition. It's very important to look at the anatomical structure, and we prepare microsections using microtome. The blade has to be sharp so that produce a nice microsection, and then follows staining with different agents to have contrast of the anatomical structure. And then after preparing the microslide, we clean it, and then we take image using a microscope attached with a camera, and then we use different softwares to quantify the anatomical structure. So we look at the color variation, the anatomical structure, and it's different from one species to another. You see the cell size differences here and the color differences. And this is Phyderbia, this is Boswellia, and this is Juniperus process. By looking at the anatomical structure, we need to know which one is a true ring, which one is false ring. So when you look at cross-section of the wood, you would see that it has light and dark color. The lighter part is formed during the early part of the growing season. In this condition, it grows faster, it's favorable, and the cell size is larger. That's why it appears light colored. Towards the end of the growing season, growing condition is tough, so the cell size is small, and that's why it appears darker. So one ring is composed of the light and the darker part. This is within 1 year. But when you look also between years, you would find narrow and wide rings. The narrow part of the ring is formed during drought years, mostly, and the wider part during moist years. After identifying what characterizes the ring of different species, we mark the ring boundary and we measure the ring widths using this instrument. It's a semi-automatic link tab. It has a software TSAP to process the measurements. So we measure different trees and we find patterns of wide and narrow growth rings. And it's important to measure many trees so that we have enough samples from a site. And after measurement, we do cross-dating. That means synchronizing the pattern of narrow and wide rings. This is a very important part in dendrochronology. If variation in the rings is influenced by similar climatic conditions, the patterns of narrow and wide rings between cores from the same trees and between different trees would match. Then we can assign the exact year of formation of each ring. After pattern matching, we remove the non-climatic events like age trend using statistical procedures. Once you have the chronology, you can use it for different purposes. First, to reconstruct climate data. It can be, what we have is about 30-40 years of climate data. So we can build a relationship of 30-40 years with the chronology, and we can reconstruct hundreds of years of climate data depending on the length of the chronology we have. Also, the length of the chronology can be extended by collecting samples from, for example, museums or old church to thousands of years. By doing that, in Europe, for example, they have more than 11,000 years of chronology. That means 11,000 years of climate information. The other advantage of this is we can use to determine the age and diameter relationship. For example, this is a juniper procera from Ethiopia, and it's 200 years. Look at the diameter. So you cannot tell just by looking at the diameter the age of the tree. This is Phyderbia, and it's 30 years. We can compare different species, and we can tell which species is resilient to drought, which species do we plant if the climate is getting dry, so we can characterize the species like that. Climate smart trees for climate smart agriculture. And restoration of degraded landscapes. And this is also the cheapest and easiest way of measuring to quantify the role of trees and forests in climate change mitigation and adaptation, like how much carbon dioxide is sequestered by trees and forests per year or during their lifetime. So by looking at the past, by understanding what has happened in the past, we can understand what's going on today to better prepared for the future. And also we develop capacity of African students. We have many MSc and PhD students visiting the lab because this technique is new in Africa. Given the diversity of environmental questions that can be addressed using dendrochronology, there is a need to expand the science in Africa, to fill in the huge data gap and limited scientific capacity and facility, to enhance African countries' ability to make evidence-based policy decisions that benefits livelihood and the environment.